I'm back again. I am Jessica Isimbahon, and today I will going to discuss another topic. This all about microbial structure. Don't forget to like, share, and subscribe. Microbial cell structure are quite varied, with some such as neurons being longer than they are wide, and others such as parenchyma being equidimensional. Some cells are encased in a rigid wall, which constrains their shape, while others have a flexible cell membrane and no rigid cell wall. The size of cell is also related to their functions. Eggs are very large, often being the largest cell an organism produces. The large size of many eggs related the process of development that occurs after the eggs is fertilized. When the contents of the egg, termed zygote, are used in rapid series of cellular divisions, inheritance trust fund of energy is used. The cell wall is located outside the plasma membrane. Animals shown in lack a cell wall. Many protistants also lack cell wall, using variously modified cell membranes who act as a boundary inside of the cell. The nucleus occur only in eukaryotic cells. Also location for most of the nucleic acids, cell mix such as DNA and RNA. Danish biologist Joachim Hammerling carried out an important experiment in 1943. His work showed the role of the nucleus controlling the shape and features of the cell. DNA is the PC carrier if inheritance, and with the exception of plastid DNA, cpDNA and mDNA, found in the chloroplast and mitochondrion respectively, all DNA restricted to the nucleus. Cytoplasm was defined earlier material between plasma membrane, cell membrane, and the nuclear envelope, fibrous protein, occur in cytoplasm. Referred as the cytoskeleton, maintain the shape of cell as anchoring organelles, moving the cell and controlling internal movement of structures. Cell cycle is the sequence of growth, DNA replication, growth and cell division that all cells go through. Cell movement is both internal, referred to as cytoplasmic streaming, and external, referred to as motility. Internal movements of organelles are governed by actin filaments and other components of the cytoskeleton. Prokaryotic cell division. Prokaryotes are much simpler in their organization than eukaryotes. There are a great many more organelles in eukaryotes. Also more chromosomes. Usual method, prokaryote. Cell division is termed binary fission. Eukaryotic cell division is more complicated, although the same processes of replication, segregation, and cytokinesis still occur. Mitosis is the process of forming generally identical daughter cells by replicating and dividing the original chromosomes, in effect making a cellular xerox. Mitosis deals only with the segregation of the chromosomes and organelles into daughter cells. Prophase is the first stage of mitosis process. Chromatin condenses, DNA replicate during interphase. Nuclear envelope dissolves, centrioles, if present, divide and migrate, and kinetochore fibrils form and spindle form. Metaphase follows prophase. The chromosomes, which at this point consist of chromatids held together by centromeres, are separated to opposite poles of the respective spindles. The nuclear envelope reforms. Chromosomes uncoil into chromatin form, and the nucleolus, which had disappeared during prophase, reform. Anaphase begins with the separation of centromeres and the polling of chromosomes. We call them chromosomes after the centromeres are separated to opposite poles of the spindle. Telophase is when chromosomes reach the poles of their respective spindles. The nuclear envelope reforms. Chromosomes uncoil into chromatin form, and the nucleolus, which had disappeared during prophase, reform. Structure of antibodies. Structurally, the antibodies, immunoglobulins, are composed of 4 protein chains arranged in a distinctive pattern. Each molecule has 2 long chains of heavy molecular weight protein, H chains, and 2 short chains of light molecular weight protein, I chains. The chains are linked by sulfur bands at the outer end of each arm of the antibody molecule. A specific amino acid sequence exists. This is where the antibody molecule reacts with the antigenic determinant that provoke its production. The combining site is known as the Fab region. The most common antibody molecules have 2 Fab regions and are said to be bivalent, having 2 combining sites. The remaining portion of the antibody molecules is called the Fc region because it can be crystallized. Its amino acid content is relatively constant and characteristic for each class. This portion of the molecule activates the complement system and encourages phagocytosis. When the antibody molecule reacts with antigen, the 2 surfaces fit together like the pieces of a jigsaw puzzle. This recognition is exquisitely accurate and accounts for the extreme specificity of antibody. Classes of antibodies. 5 classes of antibodies, immunoglobulins, are produced by the plasma cells. The first class, called IgE, is the major component of the primary antibody response in adult humans and is the first antibody to appear in the immune reaction. It is composed of 5 units joined by a joining chain. IgE cannot diffuse through cell membranes and is found almost exclusively in the blood because of its many binding sites. It is more reactive with antigens than are other kinds of antibodies. Second is IgG. This antibody is the most common in the bloodstream and is found in many secretions such as spinal, synovial, lymph, and peritoneal fluids. IgG crosses the placenta and protects the fetus and newborn. IgG therefore provides a natural type of passive immunity. Also forms in the primary antibody reaction after a large amount of IgM has already formed. IgG has the typical antibody structure of 4 protein chains. The third class of antibody is IgA. This is found in external secretions such as those at the mucosal surfaces of the respiratory, gastrointestinal, and urogenital tracts. It is also present in the thirst, saliva, bile, urine, and colostrum, and it is transferred in the breast milk. IgA consists of 2 antibody units held together by a G-chain and secretory component. IgA resists infections at the body surface. The fourth class of antibody, IgD, is found in extremely small quantities in the serum. IgD is found on the surface of B lymphocytes and is believed to be an antigen receptor at this location. IgM is also believed to be a receptor. The final kind of antibody, IgA, occurs in minute concentrations in the serum and is important in hypersensitivity reactions, especially the anaphylactic reaction.